Hello young guys, you are welcome in the zoology class organized by Society of Career Technology and Jan Vikas Mahasabha. Today we will talk about cell cycle. You have to differentiate between cell cycle and cell division. Actually, in the life cycle of a cell, there are two stages. One phase when cell is dividing, and second is the interphase when the cell is not dividing. So both things taken together make the cell cycle. And the interphase, in which cells are not dividing at that time, includes three stages: G1, S and G2 phase. G indicates gap or growth. So at this time, G1 and G2, there is a formation of RNA and protein. In S stage, DNA synthesis takes place, and then sometimes in the G1 stage, a G0 stage has been also observed where the growth of the cell is arrested for some time. Then there are two crucial steps. One which switches on the mitosis or cell division phase. It is switched on by M cycline along with CD-C2 kinase. It switches on the mitosis. Similarly, the synthesis of DNA is also switched on by a G1 cycline along with a CD-C2 kinase. So there are two stages: interphase, which includes three stages, G1, S and G2, and sometimes G0 also, and the M or mitosis, where cell division takes place. Now one very interesting thing is that the chromosomes are made up of DNA molecule, DNA and a stone, and DNA contains two helices arranged in a double helix fashion. So what happens at the time of cell division? The two helices separate from each other. But you see, after some time, both these ends cause an inhibition in further opening of this. So what happens? When this stage occurs, the two strands are separating, and here there is a twist. Here a cut takes place by helicase enzyme, and it rotates this way and then joins again. This is done by helicase and topoisomerase enzyme. So this is one very important step. Second important step is that when the two DNA helices separate from each other, and DNA replication is semi-conservative, each helix acts as a template, and parallel to it complementary nucleotides are brought and attached. But there is two crucial steps here. One strand is called leading strand and second is called lagging strand. In DNA, the nucleotides have two ends. This sugar, it has three ends attached to a phosphate, and here the 5 carbon attached to a phosphate. So there is a 3 end and a 5 end. So here in one strand, leading strand, this is 5' end, this is 3' end. Here the complementary strands will be brought in one stroke. But one more thing, the DNA polymerase cannot act from the terminal ends. It will act from a primer having both strands together. So every time a primer is attached here in which both strands are there, a small piece, then DNA polymerase starts adding fresh nucleotides adjacent to it. But in the another strand, lagging strand, because the complementary strands will be attached only in 5-3 direction. Here the 5-3 direction, suppose it is opening from this side. So what will happen? In this side, a small primer will be attached and some fresh nucleotides will be attached, a small piece will be formed. Then again in a small area the two strands will separate. In another piece, again a primer will be attached, and to this primer some more nucleotides are attached. So in one of the strand, in the lagging strand, the complementary nucleotides are attached in pieces. They are called, on the name of its discoverer, a Japanese scientist, they are called Okazaki fragment or Okazaki fragment. So in one of these strands, the nucleotides are attached in pieces which are called Okazaki fragment. So this was about the interphase. And now let us come to the dividing stage. The division includes both division of nucleus or karyokinesis and division of cytoplasm or cytokinesis. The division of nucleus or karyokinesis takes place by 4 methods. The first one is amitosis. Here, of course, there is division of a nucleus, but the changes taking place at chromosome level are not visible. In some primitive prokaryotes and primitive organisms, this type of division takes place, where the nucleus, you can see here, constricts into 2 dumbbell-shaped structures and then they get separated. After that, cytoplasm also divides and they form 2 daughter cells. In some of the sporozoan parasites, this type of division takes place, where you can see here the nucleus breaks into many pieces, and around each piece some of the cytoplasmic material gets accumulated. They form a small cell-like structure called spores. When all these spores are formed, the parent cell membrane is destroyed and all these new spores come out of the cell. Now we will talk about mitosis and meiosis. So during mitosis, the chromosome number remains the same, but in meiosis the chromosome number is reduced to half. In mitosis, what happens? Before starting mitosis, in the interphase stage, inside nucleus, the chromatin material is present in the form of a network-like structure. As the cell division starts, during prophase, 4 changes take place simultaneously. First, the centrosome divides into 2 and both develop a repulsive force. They start separating from each other. Later on they form the 2 ports. The nuclear membrane also starts dissolving. The chromatin material breaks into distinct thread-like structures called the chromosomes, and the nucleolus also starts disappearing. So all these 4 changes make the prophase. Later on, in metaphase, what happens? A repulsive force develops in between the pole and centromere of the chromosome. So here this pole also repels the chromosomes towards the opposite side, and here this pole also repels the chromosomes. As a result of equal repulsive forces from both sides, from both poles, all these chromosomes arrange in the middle at equatorial plate. So this is called metaphase. Till now, all these chromosomes contained 2 chromatids attached with a single chromatid. Now, a division of this chromatid takes place. So 2 chromosomes are formed each having a single chromatid. Now, a repulsive force again develops in between these two centromeres. So just see the irony. On one hand, the poles are also repelling the chromosomes. At the same time, a new repulsive force develops in between the centromeres of the two chromosomes. So they also start separating and moving towards the two poles. So make a V or J shape. This is called anaphase. Now in the fourth stage, telophase, what happens? As the two repulsive force, this one and a repulsive force in between these two, become neutralized, become equal, further movement of chromosome towards the poles is stopped. And all changes taken place in prophase now take place in just reverse order. So what happens? Here the nuclear membrane was dissolved. Here new nuclear membranes are formed around the two groups of chromosomes. A new nucleolus starts developing, and the chromosomes again intermingle with each other to form a chromatin network. So two daughter nuclei are formed, each having the same number of chromosomes. This is called telophase. After that, during division of cytoplasm or cytokinesis, it takes place by two methods. In animal cells, what happens? It is very simple. Here, an animal cell in which two daughter nuclei has been formed, a constriction or furrow develops in the middle. For example, if you take a balloon and in the middle part you just compress it by your fist. So the two ends start coming close to each other, and then they unite with each other. Two daughter cells are formed. In plant cells, outward to the plasma membrane there is a cellulose cell wall also, so furrowing is not possible. Here, from the vesicles of Golgi body, here some membrane material start depositing in the middle, called the cell plate, and it develops further. Two membranes are formed, so two daughter cells are formed. So this ends the cytokinesis. Thank you very much.